So section 1.3, part 2 of genetics. In part 1, we looked at gene mutations. So now we're going to focus on chromosomal mutations. Chromosomal mutations. So this is a mutation of all or part of a chromosome. So instead of looking at one or two bases in a gene, we're now looking at all or a part of a chromosome. This usually involves large segments of DNA and therefore can have greater impact than smaller gene mutations. There are 4 types of chromosomal mutations that involve part of a chromosome. So all 4 of those are circled in the bottom and are represented by a chromosome diagram as well as a little kind of stick figure diagram to help you visualize this. So all 4 of these involve not all of the chromosome, but part of it. So we'll get into full chromosome mutations after, but this is just dealing with a portion. So to start off, deletions is the first one, and essentially a segment is deleted. So a portion of the chromosome is gone. In a deletion, part of your chromosome segment is lost. So in our example shown here, it has segments listed alphabetically, A, B, C, D, E, so on till T. So in a deletion, part of it is gone. This part here is essentially lost. So it goes A, B, G, H, so on down to T. In translocation, the second one, it's not a full-blown deletion, but instead a segment from one chromosome, in this case the green chromosome, is transferred to another chromosome, which is shown in blue. So here, section A to F, so alphabetically, gets attached to the blue chromosome, which is numbered numerically. So you're not losing any genetic information, but it's not found in the right place. Okay, so it's kind of represented by if you had 2 smiley faces, if the one smiley face's head got added onto the second smiley face. So you can see that he now has 2 heads that are from different chromosomes, so they look different. Okay, in a duplication, a segment from one chromosome is transferred to its homologous chromosome pair, which gives duplicates of some genes. So we have all the letters working with homologous chromosomes here, so remember that. But we have 2 copies of gene A, 2 copies of gene B, 2 copies of gene C, 2 copies of D, and E. So that's what's shown in kind of that circled part. So we have 2 of everything like normal, but the difference is it's now on one chromosome, which has 2 copies. So that one chromosome has 2 copies of, say, 5 genes, but the other one is now missing a segment shown by kind of this blank here of those 5 genes. So this is represented by, again, you've got a smiley face, but the head was duplicated and put on top. And so the other one would technically be a black stick figure with without a head on it. So it's showing that one loses a portion and the other one gains it, but that gain is exactly the same. So you've got a duplicate. Okay, in inversion, a segment of the chromosome is inverted, so that means it's flipped. So the segment rotates 180 degrees. Segment CDEF, which is shown in the middle there, is flipped. So it becomes FEDC. So instead of being A, B, C, D all the way to T, it's now A, B, and then it goes F, E, D, C, G, so on till T. So all of the genetic material is here still, but it's not where it would normally be or where you'd expect it to be. So chromosomal mutations. So if we look at the first row, this represents a deletion. So you can see that B here has been removed from here. So segment B has been lost. In a duplication, which is the second row, B has now been doubled. So we can see that here. Inversion, which is the third one, the chromosome gets twisted on itself and gets put back together in the wrong order. So you can see that it goes AED, where it should go ABC. So B and C have been flipped to the second arm of this chromosome. In translocation, part of one chromosome, which is the last example, is shifted to another chromosome. So remember, this is dealing with two separate chromosomes. So it's being symbolized here by if ABCDE was the red chromosome and GHI to L was the blue chromosome, you can see that a portion of red is now with blue on both of them. So all the information is there, but not necessarily where you would expect it to be. Okay, so let's look at an example of this. Fruit flies are great for genetic experiments because they only have 4 pairs of chromosomes. They also breed quickly, so you can look at the next generation easily. Most genetic research, was originally done on fruit flies and still continues to be researched to this day. David Suzuki, for example, who is a famous environmentalist from Canada, actually did a lot of his research in university in genetics, and he was actually the first person to sequence the genome of the fruit fly. So for our example here, fruit fly with legs in the place of antennae. So essentially it's got legs where the antennae would usually be. So this mutation involves moving a controller gene that tells the fruit flies where to put the legs. Basically, the mutation causes it to be the legs to be in a different position. So you end up with movement of a controller gene that tells the developing fruit fly where to put the legs. The movement of the location of the legs means we have a translocation. So the instruction that make the legs are there, but they're not placed where they should be. As a result of the instructions being placed in the wrong place on the chromosome, this means that the legs get placed in the wrong place physically because development is occurring in the wrong order. DeGeorge is a human example of a deletion chromosomal mutation. So, example, DeGeorge syndrome on chromosome 22 is the loss of certain genes, which is a deletion, which results in heart defects, poor immune system, a cleft palate, as well as some other abnormalities. So there are a lot of things that come along with this one section of chromosome 22 that is missing. So this individual, although missing this segment of chromosome 22, is still functional in a lot of ways, but because there's a relatively significant number of genes missing, you end up with some regulatory stuff being turned off or missing. So this example tells us which genes are being regulated by this segment on chromosome 22 because an individual with DeGeorge is missing and has a significantly shorter chromosome 22 when compared to somebody without DeGeorge syndrome. So in addition to a part of a chromosome mutation, we can end up with a whole chromosome mutation. So sometimes if errors occur during sperm and egg development, which is meiosis, there may be an entire extra chromosome added or an entire chromosome may be lost. So let's kind of talk about this a little bit more on the next slide. So an example of a chromosome being added, an entire chromosome, would be Down syndrome, which is a genetic disorder where there is an extra chromosome 21. The official name is trisomy 21. Tri means 3. The extra chromosome is a result of improper cell division. So when chromosomes go to line up along the central line during meiosis and pull apart to opposite sides, they don't get pulled apart properly. So if we look at the karyotype, which is here, so notice the 21 circled in red. So the karyotype of an individual with trisomy, we can see that there are 3 copies of chromosome 21, where there should only be 2. So this is what results in an individual with Down syndrome's physical, cognitive, and personality traits that seem to be tied with a duplication in chromosome 21. So let's talk about why mutations can actually happen. So causes of mutations. There are a ton of mutations that can potentially happen without actually going on to cause a problem. So keep this in mind. Mutations can occur naturally during DNA replication and cell division. The human body has a system in place that will often fix errors in DNA replication or destroy cells working abnormally due to mutations. So mutations occur naturally all the time because DNA contains a lot of information that is being copied. So you can imagine that if you were trying to copy down a ton of information, you would make the occasional mistake. But there are factors in the environment, in addition to these random mutations, that can make it more likely for a mutation to occur. So these are what are called mutagens. So mutagens are substances or factors, i.e. virus, cigarette smoke, X-rays, that can cause mutations in DNA. If a mutagen causes a mutation that causes cancer, it's called a carcinogen. So how many of these have you been exposed to? So I'm going to show you some images here. Have you gone outside? Then you've been exposed to UV radiation. Have you ever had an X-ray for a broken limb or at a dental office or at airport security? An X-ray actually has a higher dose of radiation than that of the sun, but the sun you're going to be exposed to no matter what. Various chemicals in cigarette smoke, either through smoking it yourself or through secondhand smoke. Have you had foods with preservatives in them? Usually, nitrates are often used to preserve a lot of processed meats. If you burn something while cooking or barbecuing, this can cause mutinogenic chemicals in foods to form. Benzoyl peroxide is a super common ingredient in acne products. Infectious agents such as HPV, human papilloma virus, is a sexually transmitted virus and is linked to cervical cancers, among others. And then last, there are some bacteria that can spread through contaminated food that can also be considered mutanogens because they can cause mutations within the cell. So most of the time, our body, as stated before, deals with the effects of these mutagens and nothing bad occurs. However, sometimes this can lead to cancer. So cancer is caused by several mutations that occur in the cell cycle. So this is again going back to grade 9, causing cells to reproduce out of control. This results in large, deformed cell masses that we call tumors. If a tumor goes undetected, pieces of cancerous tissue can break off and move through the bloodstream, landing elsewhere in the body and starting new colonies of tumors. This is why early detection and removal of tumors is crucial. So let's look at what this spreading looks like. So in the first image there, we can see that cells are growing and forming a benign tumor in the epithelium. The yellow line is the basal lamina, so it's like a barrier for your cell type. So cells break through and get into the bloodstream by invading the capillaries, and they travel through the bloodstream to elsewhere in the body. In this case, we're looking at the liver. It adheres to the blood vessels walls in the liver and then travel through the blood vessel wall into the liver itself. From here, the cells quickly grow and divide in the liver. So basically, you're going from skin cancer to liver cancer relatively quickly, unfortunately. So if a tumor is not cancerous and not spreading, it's said to be benign. If a tumor is cancerous, it's called malignant. If a malignant tumor is spreading, it's said to have metastasized. So when you end up with a primary source of cancer that is spread to other parts of the body, this is the idea of metastasized. So this occurs by cancerous cells breaking off and going through blood vessels to other parts of the body. A cancer cell is different from a normal body cell in several ways. Cancer cells have abnormal structure, and they are often bigger, have too many or too few chromosomes, and a cancer cell may have multiple nuclei in it. So basically, they're reproducing so quickly that there's tons and tons of errors, and none of them are being corrected. Cancer cells can produce chemical messages that tell the body to provide it with blood vessels and provide the cancer through those blood vessels with nutrients. This can cause strain on the body. They draw nutrients from elsewhere in the body by signaling the body to bring the cells more stuff, and that is what causes the strain on the body. So this is usually why individuals with cancer are weaker and in general can often look malnourished. It is not that they aren't eating enough or eating healthy, it's that the cancer is taking away a lot of the nutrients from them. Cancer cells are misshapen and often do not function properly. For example, if it's a cancerous skin cell, it doesn't care about being skin or a skin cell, it just wants to grow and divide and continue to grow. Cancer cells multiply rapidly as often as 24 hours. So by pulling in all those extra nutrients, they are able to grow and divide a lot faster than a normal cell would. All right, so this brings us to the end of section 1.3. So you now have some review questions, which are on page 60 as well as here, to complete.